Welcome to this presentation on a simplified, template-driven strategy for rapid HPLC method development in pharmaceutical analysis. Research shows that people learn new processes more effectively when following a template. Today, we will introduce 3 HPLC method templates that represent the major quality control methods for small molecule drugs. These are achiral, reverse phase, UV-based methods designed to accelerate early phase method development and support CMC activities for NCE projects. Our strategy is categorized into 3 distinct templates based on complexity and purpose. First, the fast LC isocratic method used for potency and performance, which is considered very easy. Second, the generic broad gradient method, suited for high throughput screening or in-process control, which is relatively easy. Finally, the stability indicating method involving multi-segment gradients for ICH assays and impurity methods, which is the most challenging. This 3-pronged approach improves speed, consistency, and decision-making by aligning each method with its intended goals. To recap, we focus on fast LC isocratic methods, generic broad gradient methods, and stability indicating multi-segment gradient methods to streamline the CMC development process. Let's look at template 1, the fast LC isocratic method. Using a short Waters X-Bridge C18 column, we can achieve run times of less than 2 minutes. This is ideal for dissolution testing and content uniformity. In this case study of an AKT inhibitor, the method was developed and pre-qualified within hours, showing excellent performance for content uniformity with a K-prime of about 1.0. Template 2 covers generic broad gradient methods, typically used for starting materials or in-process controls. These methods require minimal adjustment. For example, using an ACE C18 column and a 15-minute gradient from 5 to 95% organic phase, we achieved a purity result of 97.8% for a starting material. The top chromatogram shows a standard mix spiked with precursors, while the bottom shows the actual release sample. For stability indicating assays of complex APIs, we use template 3 multi-segment gradients. Since impurities and degradants often resemble the API, we use a shallow gradient segment to maximize resolution around the main peak. A steep gradient is then used to elute strongly retained components, while a low-strength segment may be added to retain polar impurities. When dealing with APIs that have multiple chiral centers, such as this AKT inhibitor with 3 centers, the method must separate the API from multiple pairs of diastereomers. While enantiomers require chiral methods, our reverse phase approach focuses on resolving these diastereomers and process impurities to ensure product quality. In this case study, we developed a 42-minute primary regulatory method using a C18 column and acetonitrile. We also developed an orthogonal secondary method using a phenyl column and methanol for conformation. The primary method successfully separated the API from several diastereomers, revealing a purity of 95.4% for the GLP tox lot. To further optimize, the method was redeveloped for UHPLC using a 1.7 micrometer column. This reduced the runtime to 27 minutes while providing baseline resolution for 4 pairs of diastereomers. As a result, the first GMP batch of the API was confirmed to have an impressive purity of 99.5%. For extremely complex pharmaceuticals, such as the scribbled oral tablet containing 4 APIs, we use high-resolution UHPLC. This chromatogram shows the separation of a 60-component mixture. A 5-segment linear gradient was developed and validated using quality by design software to ensure the separation of all APIs and their various impurities. In conclusion, the 3-pronged template strategy provides a practical and structured starting point for HPLC method development. By focusing on fast isocratic methods, generic broad gradients, and multi-segment stability indicating methods, we can effectively meet the goals and attributes of modern pharmaceutical analysis. For more detailed information, please refer to these key resources, including HPLC and UHPLC for practicing scientists and the various LCGC North America articles that detail this template-driven approach. Thank you for your attention. We are now open for any questions regarding these HPLC method development strategies.